Coming to antibody-mediated rejection, antibody-mediated rejection can be classified into acute or chronic antibody-mediated rejection. Based on that, we have some histological evidences for antibody-mediated rejection, and there must be three things that should be present. Three, like you can say, a criteria. There should be evidence of histological changes. There should be presence of antibody-mediated vascular injury, and presence of DSA, donor specific antibodies. Similarly, same is for chronic antibody mediated rejection. Criteria is histological evidence, antibody mediated vascular injury, and presence of DSA. So basically what we are talking is like what happens histopathologically. If you see the slide of a graft, so what you are going to observe, starting from the very basic, what will be the finding in what will be wrong with glomeruli. Second thing which we see is capillaries, and any evidence of thrombi, or any other form of injury, or any injury. So how we classify that based on this, that in acute ABMR histopathology, we ask such that there will be glomerulitis, there will be pericapillaritis, like there will be inflammatory injury, there will be inflammatory cells, inflammatory injury. It is going to be acute. So in acute inflammation, we know that there is migration of the cells with very corresponding histological compartments, and there can be TMA as well, microthrombi can be there. So this is acute. So chronic ABMR, we said that it's having histological evidence, and there might be some changes, immunofluorescence changes, signified by the presence of C4D staining. Histological evidences, we know that, like we said previously, three changes. Histological evidences, antibody-mediated vascular injury, or any other sort of injury, and presence of DSA. So how it's going to be differentiated from? It's going to be differentiated from this, this acute antibody-mediated rejection. So based on that, the histological compartment involvement shows some degree of chronicity. How? There is the glomerulitis become glomerulopathy, capillaritis become capillulopathy, and there is some intimal fibrosis as well, and of course presence of C4D stain in 50% of the cases, and greater than 10%, we can say, of the cases on IF. Glomerulopathy in chronic ABMR is called transplant glomerulopathy. And that's because the GBM there is double contouring of the GBM, thickening of the GBM, glomerular basement membrane thickening. That's because of the fact that there are some subendothelial deposition, right? So we say double contouring. But this double contouring is not just specific for for transplant glomerulopathy. There are some other differentials. For that, we'll come to that later. But besides that there is pericapillary fibrosis or intimal fibrosis, right? So this was all about antibody mediated rejection. Coming towards this differentials of transplant glomerulopathy. So it can be present in MPGN, it can be present in IgA, can be present in some other autoimmune condition, and even in diabetic nephropathy as well. So these are rare different. So continuing the Banff classification, so far we have discussed the acute ABMR and its type and the type of rejection that's occurring in it and what are the changes. It's coming to the category 4, which is T cell mediated rejection. So T cell mediated can be classified into active, acute, then chronic, which is subclassified into 1, 2 is classified into 2A and 2B, and then 3. Same goes for the chronic, which is classified into 1, 2A, and then 2B. So coming to that, class 1A or class 1 or subclass 1, in which we say that's further classified into 1A and 1B. So as we said that there is some degree of tubulitis and tubular injury, same goes for this in TCMR as well. There is interstitial inflammation in both 1A and 1B. But how 1A is different from 1B? It is different from 1B based on the tubulitis and the number of cells. In 1A, the cells are usually less than 10, the lymphocytes. The main cells are lymphocytes, and here it can be equal or more than 10 lymphocytes in 1B. Similarly, if we come across the subclass 2, so 2 is classified into 2A and 2B, and there is some degree of vascular injury, vasculitis, or we can say vascular injury. Okay, so if it's like, if this is the vessel and we divide it into 25 and the rest is divided into 25, like if we divide it into three portions, if it is involving 25%, up to 25%, then we say it's 25% arthritis. If it's involving like greater than 25%, like up to 50% maybe, so we say that this is 2B. But if it's involving the whole, so then we call it class 3, which is called subendothelial arthritis, and it's transmural because all the segments are involved. So this is 3. Same is the case with the chronic, based on the presence of fibrosis. 1, 2A, 2B. 25%, greater than 25%, and complete IFTA.